Hello everyone and welcome. My name is Lauren Etsitty and today I'll be giving a presentation about the 10x Visium tissue quality control procedure conducted at my summer internship held by NIH at the Columbia University Irving Medical Center and the New York Genome Center. Senescent cells were discovered in the 1960s when Leonard Hayflick and Paul Moorhead noticed the cells associated with wound healing, also known as fibroblasts, stopped reproduction at a limit of 50. This was later coined as the Hayflick limit. These cells were then labeled as senescent, meaning the cell reproduction discontinued, the cell continued to perform normal cell functions instead of dying. It was later found the accumulation of senescent cells increased with age. Considering senescence is a recent discovery, there is much to be studied. For this reason, the National Institute of Health's Common Fund has created the Cellular Senescent Network, also known as SenNet, to identify differences in senescent cells across the body, various states of human health, and across different lifespans. To answer the questions of how, why, when, and where senescent cells form, and its beneficial and detrimental effects on the human body. Specifying the three main focuses, Columbia University's Tissue Mapping Center and the New York Genome Center is building an atlas of senescent cells in healthy individuals spanning from 20 to 80 years old. The workflow constructed for this research is represented in the following chart. First, we seek tissue samples from the brain, spinal cord, and hippocampus, while noting non-pathological aging characteristics, which include young, middle-aged, and elderly persons. Next, these tissue samples are used in the following processes. 10x Visium to generate genome-wide spatial transcriptomic data, sRNA sequencing to generate cell-type gene expression profiles, 4i with P100 antibody panel to define cell types and pathways. All of these three procedures work in an effort to build the 3D molecular atlas of senescence in human tissue across the lifespan. In this process, additional samples are saved for validation and imaging processes, showing an aim for. Once this atlas has been created and the blood from CSF is extracted from adults and gone through the CASP analysis process, we can begin our data analysis core, which includes data processing, characterization, generating maps, integrating tissue and SASP data, and determining biomarkers for senescence. Lastly, this data will be used for image and validating purposes as well to create accurate models and permutations. Within this overall workflow, my summer internship focused on the 10x Visium quality control process, which included tissue sectioning, H&E staining, image processing, and annotations. As mentioned before, Columbia University's Tissue Mapping Center is conducting research on the brain, which includes the dorsolateral prefrontal cortex and various Brodmann areas, including BA44, BA46, BA9, and the motor cortex in regions BA4, the hippocampus, and the spinal cord in the cervical and lumbar regions. Once the tissue sample is sectioned and adhered to the glass slide, we can begin the hemotoxin and eosin staining process. After taking out the glass slide from the negative 80 degrees Celsius freezer, we first begin this process with a methanol fixation method, where the sample remains submerged in methanol at negative 20 degrees Celsius for 30 minutes. Next, we apply isopropanol on top of the sample for tissue dehydration. Next, hemotoxin is used to stain the acidic structures such as the heterochromatin and nucleoli. Then a bluing buffer is applied to increase detail in the nuclear staining. Lastly, an eosin and tris acetic acid solution is used to stain the basic structures such as collagen, elastic fibers, muscle fibers, and red blood cells. Once the staining procedure is complete, glycerol is then used to mount the glass cover to the glass slide to seal the H&E stain. Once the H&E stain is complete, it is time for image processing. In this process, you will use an epifluorescent microscope and the ZenBlue 3.4 software to look over the glass slide with the stained tissue sample, and note any freeze fracture, folds, and/or tears in the tissue sample for later annotation analysis. Once a tissue sample is deemed to have clear morphology, it can move along in the process to be imaged. These images are then saved as a raw CZI and stitched, then saved as a JPEG, which is best for tissue annotations. Today, we'll be looking at the three types of stained and imaged tissues in the motor cortex region, hippocampus, and spinal cord. The annotation method for any motor cortex tissue includes securing the white matter regions, identifying granule cells, identifying pure metal cells, which can be small or large, and identifying the regions with different cell density. For example, today we'll be looking at how this method is used on a BA46 tissue sample for a middle-aged male. Based on the annotation procedure, the white matter will first be highlighted in pink. Then surrounding the white matter, the pyramidal cells are identified and circled in yellow. Depending on where the smaller and larger pure metal cells lie, layer 5 will be along the trajectory of the larger pure metal cells, and layer 3 will be along the trajectory of the smaller pure metal cells. Note, there should only be pyramidal cells in layers 3, 5, and 6. With this considered, layer 6 can be filled in because it is the remaining region in between the pink matter and layer 5 that consists of random cell types and pure metal cells. Then layer 4 can be defined as a layer in between 3 and 5 that consists of no pure metal cells and has neurons with very small cell bodies. Layer 2 and layer 1 can then be deducted to the area furthest from the pink matter and can be differentiated by cell density, with layer 2 being more dense than layer 1. And this is our final annotation of a BA46 tissue sample. Next, we will look at the annotation method and example for the hippocampus. In the annotation method, we will first identify the granule cells and mossy cells which remain in this region. Next, we will take note of the drastic cell density changes among the tissue sample. Then we'll identify areas where the tissue has cell-free areas and/or dendrites. Once again, we will identify the pyramidal neurons and white matter similar to the motor cortex region. For today's example, we'll be looking at how this method is used on a hippocampus tissue for an elderly female. Using the annotation method procedure, the white matter is the darkest stain on the sample and first highlighted in pink. Next, the cornu ammonis 4 is highlighted, which contains the granule and mossy cells. Then the gentite gyrus granular is highlighted, which consists of density-packed granule cells. Next, the dendrite gyrus molecular is a region with very few cells and/or a cell-free layer consisting of dendrites. Next, the cornu ammonia stratum lacunosum molecular is highlighted and identified due to its moderate density of nuclei. Next, the cornu ammona radiatum is highlighted and identified due to pyramidal cells and sparse nuclei density. Lastly, the cornu ammonia pyramidal is identified due to its pyramidal neurons and highlighted. Lastly, we look at the annotation method and example for the spinal cord. For this annotation method, the general regions of the spinal cord are identified using the shade variation provided by the H&E stain with the butterfly-shaped region of the gray matter. In today's example, we'll be looking at the spinal cord lumbar tissue sample on a middle-aged male. The ventral horn portion is located at the bottom portion of the butterfly shape. The ventral edge portion surrounds the outskirts of the spine under the bottom portion of the butterfly shape. The ventral medway is the region between the ventral horn and the ventral edge. The ventral latway is the region on the far right. The metlatway is the remainder of the white matter under the top portion of the butterfly wing shape area. The medgray is the middle portion of the butterfly. The dorsal horn is the top portion of the butterfly shape. The dorsal medway is the matter above the butterfly shape. The dorsal edge is the outskirts of the white matter. The black circle is the accumulation of cells in the middle of the spinal cord. These are the complete examples of the motor cortex region, hippocampus, and spinal cord lumbar annotations. For all completed annotations, they will be stored in a drive and later used for splotch annotations, which is the later workflow in the overall Columbia University tissue mapping workflow. So thank you everyone for watching. I want to give a special thank you to my supervisors, Shanice Greer and Nadia Prop, as well as the Columbia University Irving Medical Center and the New York Genome Center, and overall NIH's SenNet Consortium. Thank you.